afternoon, everybody. Today I want to talk about genomes, viral genomes. We're going to go through all the different kinds of viral genomes that exist, and then we're going to talk about how you can manipulate them to make viral mutants and use viruses for the common good. So the title of today is Genomes and Genetics. Now, when, I don't know if you know this, but in the 1940s, it was determined that the DNA of bacteria is the genetic material. Up until then, for the first 40 years or so of the 20th century, there was a lot of debate as to whether protein or nucleic acid was genetic information. Can you imagine? We can't look back and even imagine that, but there are people who lived through that. The reason why people thought proteins might be genetic material because they're more complicated than nucleic acids. Nucleic acids only had 4 bases, and there are at least 21 amino acids. But in 1944, an experiment was done here in New York City at the Rockefeller Institute, Rockefeller University. Avery, McLeod, and McLeod showed that DNA of bacteria contains genetic information. Of course, for years, people didn't believe them. They said, oh, your DNA is contaminated with protein. But that's usually what happens. Eventually, they were believed. But we didn't know what the nucleic acid was for viruses until the 1950s, when these 2 experiments shown on this slide were done. On the left is a very well-known experiment with bacteriophage T4 that was done by Hershey Chase. So you probably learned this in high school. I hope you did. If not, you should email your teachers and tell them they need to teach it, because a lot of kids I know in high school have been taught this. And this is a bacteriophage which contains DNA. I'm going to tell you the experiment again in a moment in case you don't remember it from AP Bio. And that showed that the DNA of the phage was the genetic material. On the right is a picture of tobacco mosaic virus, which you should remember was the first virus to be discovered at the end of the 1800s. It's an RNA-containing virus. And work that I'm not going to go into, done by Frankel Conrad and his associates, showed that it's the RNA of TMV that is the genetic information. What he did was to take apart the RNA and the protein. He purified them separately, and he would put the RNA into cells, and they started an infection. If you put the protein into cells, they did not. All right, so 2 really important experiments in the 50s established that DNA or RNA is the genetic material of viruses. And from that point on, we begin to do experiments to see how we can manipulate those. So let's take a look at the Hershey-Chase experiment. Famous experiment, as I said, many people are taught early on in their science careers. And what Hershey and Chase did, so here's Hershey, Al Hershey at the right in 1952, and Martha Chase was his lab assistant. He said, let's figure out if it's DNA or protein that's the genetic information of phages. So he had a bacteriophage shown in the upper left here, and he grew these in E. coli with radioactive sulfur or radioactive phosphorus. Of course, the radioactive sulfur isotopically labels the protein of the virus. When you grow the virus with phosphorus, P32, it isotopically labels the viral DNA. So he takes the viruses labeled in either way, takes your protein-labeled virus, he infects cells, and then here's the famous experiment. After allowing the virus to attach and inject its DNA into the host, he then put them in a blender to shear off the phages so no more infection would occur. And then he asked, where is the radioactivity? And he found that after he sheared off the viruses in his blender, the radioactivity was in the supernatant. And then if he let the cells incubate and let the infection proceed to the end, he found that no radioactivity was detected in the next generation of phage. So that meant that the sulfur didn't get into the cells and it wasn't incorporated into the new phages. So this blending, he used a kitchen blender to do this. And it's over here on the lower left is a picture of it. If you go out to Cold Spring Harbor out on Long Island, which is where he worked and did these experiments, you can go to the library there and see this kitchen blender. They kept it. It's like a little museum to the famous kitchen blender experiment. Really cheap piece of equipment that you can do amazing things with. All right, so that is when you radioactively label the protein. If you radioactively label the nucleic acid, you do the same experiment. You allow the phage to absorb, inject its nucleic acid. Of course, now we know it's injecting its nucleic acid, but at the time we didn't. He then asks, when you shear off the phages, where is the radioactivity? It's predominantly within the cells. And then in the next generation of viruses that are produced, the progeny phage, the radioactivity is in them. So it gets into the DNA and in the new phages. So this tells you that the DNA is the genetic information. We'll do another experiment today to prove this, to support this. But this was the key experiment. Now, science is full of serendipity. He picked the right phage. Because some phages, we now know, the whole thing gets inside of the cell, right? So what would you have thought if he did the experiment and both DNA and protein got into the cell, which is the case for many phages. So he happened to pick a phage where the DNA is injected and the protein is left outside. But eventually people would have figured it out. But in science, so much serendipity is involved. All right, so now we have DNA or RNA as the genetic material. Now, since the 50s, actually since the early 1900s, as we spoke the other day, many, many viruses have been discovered. Thousands of different viruses with a seemingly infinite complexity of shapes and genomes. But in fact, studies from the 50s to the 60s and 70s showed that there is a finite number of viral genomes. And I have picked the number 7 subway sign so that you will remember that. You can't forget it next time you go in the subway and you see the number 7, or even if you see the 1 or 2, you'll think 7 and you're going to remember the number of genomes, the 7 train. 7 different genome types. Despite having thousands and thousands of different kinds of particles, only 7 different genome types. And that's what I want to talk about for some time this morning. What are these 7 genome types and how do they work? So this is one way that you can remember basically all of virology. So any new virus that's discovered from now to whenever will most likely fall, its genome will fall within one of these 7 different types. Now, that is one way that we're going to use to kind of simplify and unify viruses. The other way is this key fact, which I mentioned in the first session, and that is you have to make mRNA from the viral genome that can be read by host ribosomes. Now, the way it's written here, I don't really like. Viral genomes must make mRNA, but I want viral genomes first in that sentence, because genomes don't make mRNA, right? An enzyme makes mRNA from the genome. But the point is, mRNA that is translatable by cellular ribosomes has to be produced from every virus genome, otherwise infection won't proceed. So there's a ribosome. Looks like a turkey, I know, but it's not. In our book, in our textbook, people have called them flying turkeys when they're flying around in the cell, but that's what the artist did, is actually a ribosome. All viruses on the planet follow this rule. We haven't found any exception, and that's because no virus encodes a translation system. If we find one day that a virus encodes everything it needs to make proteins, then the rule will change. But that's a lot. You have to make ribosomes, so you need all the ribosomal proteins, all the ribosomal RNAs, that's a lot. Then you need to have all the amino acids, tRNAs, tRNA synthetases. I doubt it will ever be found. I don't think viruses are going to have a complete translation system. So for now, we are safe in saying a key fact is that viral genomes have to make mRNA. Now, why, you may ask, so what? Why does that make my life easier? Well, you're going to see, because we can trace every viral genome to making mRNA, because that's going to be a central point in the scheme of replication of viruses. And in fact, this fact is exactly what David Baltimore did in the 60s and 70s. David Baltimore is here with the big fish. He's a Nobel laureate. He discovered a really important enzyme called reverse transcriptase that we're going to encounter today and many times throughout this course. And for that discovery, he and 2 others received a Nobel Prize in 1976, 77. But before that, he'd been thinking of ways to simplify and unify viral genomes. So he knew that viruses had to make mRNA, so he made a scheme called the Baltimore Scheme, where he put mRNA in the middle, and he said, let me see how many different genome types we can find, and then we can trace their path to mRNA. So that's what the scheme does. Now, in his original Baltimore Scheme, published in the 70s, there were only 6 different genome types. And then after he put it together, a seventh one was discovered. So the genome types are labeled 1, 2, 3, 4, 5, 6. And the seventh one is a peculiar kind of double-stranded DNA, which has gaps in it, we'll talk about today, of the hepatitis B viruses, or the hepadnaviridae. So that didn't exist when he made his scheme, so he had 6. But now we have 7. That's why I say there are 7 different genome types. So he put mRNA in the middle, and he said, let's then draw the 6 genome types, or 7 genome types, and trace how we get to mRNA. And this way you can understand immediately how every virus replicates. So all you have to do is learn this scheme, and a great deal of virology is accessible to you. So for example, let's take viruses with double-stranded DNA. Right there, that's class 1. They can go directly to mRNA. In fact, this is a rule that you should remember. It'll really help. The only kind of DNA that can be a substrate for transcription, you know that transcription is production of mRNA from DNA. The only substrate is double-stranded DNA. Not single-stranded DNA, not gapped DNA, as for the hepadnaviruses, but double-stranded DNA. So here in the scheme, double-stranded DNA viruses can make mRNA directly. Now, if you have a single-stranded DNA virus, it has to be made double-stranded first. Cannot make mRNA from single-stranded DNA. If you have a gapped DNA, you have to repair the gaps, make it fully double-stranded, and then you can make RNA. So you see now, we're beginning to see these pathways, how to get from the genome to the mRNA. Then those are 3 different kinds of viruses with DNA genomes. Yes?
What are the pluses and minuses?
We're going to get to that. Good question. We'll get to it. I think it's the next slide. 3 different kinds of viruses with DNA genomes. Then we have RNA genomes. We have double-stranded RNA. We have negative strand. We have plus strand. We'll explain what those are in a moment. And then we have another plus strand that goes through a DNA intermediate. All right. Now, again, as with double-stranded DNA being the only substrate for transcription, the only substrate for translation is a plus-stranded, single-stranded mRNA. So a double-stranded RNA, which has both the plus and the minus strands, can't be translated. But first, let's define some of these terms so that you know what I'm talking about. Then we can go back and explain all of this. So mRNA, these are conventions, which is ribosome ready. mRNA produced by transcription is ribosome ready to be translated. Then we call that the plus strand, just by convention. Has nothing to do with polarity or electricity or anything. People always ask me, does this have something to do with some fundamental property? No, they just said, let's call it the plus strand. They could have called it the minus strand, and we do everything oppositely. Plus strand is always mRNA. And then DNA of equivalent polarity is the plus strand of DNA. The equivalent polarity of DNA, all right, not the opposite. RNA and DNA, they're complements of plus strands or negative strands. Okay, it's very simple. And that will now make the chart much easier for you to understand. And I just want to warn you that just because you have a plus RNA in a virus doesn't mean that it's going to be translated, all right? All mRNA is plus RNA, but not all plus RNA is mRNA. So now in the Baltimore scheme, all the pluses and minuses make sense. For example, double-stranded DNA is plus minus DNA. Single-stranded can be plus or minus. We're showing it as plus here, and the same for the RNAs. So 7 genome types. Now, this scheme, this Baltimore scheme is beautiful. It's elegant because if I tell you the viral genome, you can tell me immediately how it gets to mRNA. You can also tell me most likely how the genome is going to replicate. So knowing only the nature of the genome, you can figure out the basic steps that have to go through to make mRNA. Again, knowing that only double-stranded DNA is a substrate for the production of mRNA, you can then figure out immediately that if you have single-stranded, you have to repair it and make it double-stranded. If it's gapped, you have to repair it and make it double-stranded. If you have a negative strand RNA virus, that can't be translated. So that virus, there are viruses that have negative stranded RNA in their particles. When that RNA gets in a cell, it cannot be translated. So one of the first things that has to happen, it has to be made into plus mRNA, because it won't be translated. Now, some viruses have, well, let's see, double-stranded RNA has both a plus and a minus strand, but in a double strand, the plus strand, which is mRNA, technically, can't be accessed by ribosomes because it's being blocked by the minus strand. So you have to actually make a fresh new plus strand from these double-stranded RNA viruses. Now, viruses with plus RNA genomes, there are two families here because there's a fundamental difference in the way they express their genetic information. This class 4 here, these viruses have plus RNA in the genome, and that's immediately translated when it gets into a cell. All that needs to be in the particle is the plus RNA, because as soon as that gets in the cytoplasm, proteins can be made. But the other class of viruses, class 6, that plus RNA is not translated. It's wrapped up in the virus particle, and the ribosomes can't access it. We'll talk about that in some detail. But basically, these viruses go through a DNA intermediate, and then mRNA is produced from the double-stranded DNA. Sounds circuitous. There are lots of Baroque things that happen in virology that we can't explain. But all I can tell you is that don't ask why it's like this, but rather these have evolved and they work. They are competitive in the evolutionary scheme of things. I always think all viruses should be plus-strand RNA viruses, as you'll see when we talk about the replication. It's the simplest scheme as far as I'm concerned, but they're not the only ones out there. All right, 7 classes of viral genomes. Here they are again, and you should just remember these. They're not hard. It's really the only ones you could figure out. The one that I think it's not here is maybe a RNA-DNA hybrid. I've never seen one of those. That could be an eighth class, but I've never seen it. Double-stranded DNA, gapped double-stranded DNA, and single-stranded DNA. Those are your DNA viruses, 3. And then the RNAs, you got double-stranded RNA. You have plus and minus single-stranded RNA, and then you have the plus RNA with a DNA intermediate. Now you may ask, why aren't there plus and minus single-stranded DNA viruses like there are for RNA viruses? There aren't. We don't know of any. The viruses that we know that have single-stranded DNA genomes package either strand, either the plus or the minus strand. So there's no distinction in viruses that have plus or minus DNA, as there is for RNA viruses. Okay? 7 classes of viral genomes. Okay, why is mRNA placed at the center of the Baltimore Scheme? A, because all virus particles contain mRNA. B, there is no specific reason. C, because all viral genomes are mRNAs. D, because all viral genomes must make mRNA. E, because Baltimore studied mRNA. Right, the answer is D, as you can see, another 100%. Very good. Second time in a row. All viral genomes have to make mRNA. That is why it is at the center of the Baltimore Scheme. Now, there are 7 genome types, but they are structurally diverse, and that's shown on this slide. In the end, there's still 7 different genome types, but for example, the double-stranded DNA can be linear or circular. That's something unusual. We don't have circles in our genomes, right? We have linear chromosomal DNA, but some viruses can have circular DNA. And this goes for double-stranded or single-stranded DNA genomes. They can be linear or circular. They can be segmented in pieces. So the virus could have one big molecule of genetic information in it, or it can have segments. We call it a segmented genome. It could be gapped, as we saw for the hepatitis B viruses. And as I said, of course, it can be plus or minus single-stranded. Can also be ambisense, which means it's both plus and minus sense. We'll talk about those kind of viruses. Can have proteins attached to them. These little orange spheres are proteins attached to either RNA, or some viral DNAs have proteins attached to them as well. The ends of the DNA sometimes are cross-linked. So here, this double-stranded DNA molecule doesn't have free 5' and 3' ends, but rather they're covalently linked. So we call that cross-linking. The practical result of that is if you melted this double-stranded DNA, which I'm sure you know you can do by heating it to a high temperature, the strands separate, and this would form a circle because the ends are linked. If you take a regular conventional double-stranded DNA without linked ends and you melt it, you get 2 single strands, okay? And then here's the hepatitis B genome again. It's got, not only is it gapped, but it has a protein linked to the DNA, and it's also got a piece of RNA stuck onto it. And why that's so will become clear later when we talk about the replication of hepatitis B viruses. So now you're probably wondering, why do we have such diversity? And I can't really answer that question. As I said earlier, diversity simply means that there are different solutions to a problem that work in different niches. However, I can tell you why we have both DNA and RNA-based viruses, whereas all other forms of life on the planet are DNA-based. That is because we think the first self-replicating molecules, or among the first self-replicating molecules to emerge on Earth a long time ago, were RNAs. So we think there was an RNA world that existed first, and this RNA world consisted first of small replicating RNA molecules that didn't code for any protein. There was no protein-based system yet. And people have shown today, actually, that you can make derivatives of RNA, and they will actually self-replicate without any enzymes present. So there was a whole RNA world present, and then we think that we had to switch to a DNA world for a variety of reasons. RNA is rather unstable. You can only make it so long. The largest RNA virus genomes we know about are about 30, 35,000 bases long. And of course, the DNA-based genomes are much, much longer. So we think one of the reasons was that DNA evolved because it's more stable, you can get bigger and so forth. It's also less susceptible to mutation, as we'll see. And we think the switch from the RNA to the DNA world came when proteins had evolved. So in the RNA world, it was initially a non-protein-based world, and then proteins evolved, ribosomes and so forth. And a protein that evolved during this time was reverse transcriptase. It's a very old enzyme that converts RNA to DNA, and we think that's the origin of the DNA world. And so we switched to DNA genomes, and most life on Earth now is based on DNA. The viruses, in fact, are relics of the RNA world, the RNAviruses. They're still around. In fact, there are a class of virus-like particles called viroids that we'll talk about way at the end of the course. These are just RNAs that don't encode any protein. And how they replicate is really fascinating. But we think those are relics of the old RNA world. They've been able to survive ever since. And of course, RNA viruses are relics in the sense that they existed before DNA viruses. So that's a good explanation for that diversity, DNA, RNA. But as far as linear, circular, segmented, double or single-stranded, plus or minus, no idea. We have no clue why all of these evolved. And as the best answer is that they all work in some form, and therefore they're evolutionarily competitive. So I would suggest you just memorize these 7 genome types. They're really easy. You can split them into DNA and RNA. And you should also be familiar with an example of a virus from each family. Now, in this course, we don't cover all viruses. It's not the kind of course, it's not a survey course. This is a principles course. I want you to understand how viruses work. And we study only certain representative viruses, and some of those are shown on here, and you should be familiar with some of them. And you're going to hear them over and over. It's not going to be hard to get familiar with them. The reason is that if I ask you a question about adenovirus, you need to know what the genome is at some point. I won't ask you an obscure virus that we haven't talked about, but one of the viruses that we use as an example. So double-stranded DNA viruses, we'll talk a lot about adenoviruses and herpes simplex viruses as examples. Single-stranded DNA parvoviruses, and that's easy to remember. If you have a pet, a dog or a cat, parvoviruses can kill them, and you have to have them vaccinated against parvoviruses. Those are single-stranded DNA viruses. The gapped ones are hepatitis B virus. Moving into the RNA viruses, double-stranded RNA viruses are reoviruses. We will talk about those in terms of disease at some point. Influenza is a great example of a negative strand RNA virus, but Ebola and measles viruses are also. And plus-stranded RNA viruses, well, I put poliovirus here. That's one of my favorite viruses. I've spent many years working on it, but Zika virus is also a plus-stranded RNA virus. And then the retroviruses, plus-stranded RNA through a DNA intermediate. So again, you should be able to deduce how mRNA is made from the genome. That all comes from the Baltimore Scheme, and how the genome is copied to make more genomes. We're going to talk about that second point in just a moment. Now, if you want to know more, there's a good website called Viral Zone. That's the URL at the top there. It's put together by a bunch of computational biologists in Switzerland. And they have divided all the viruses on the site into DNA, RNA, and reverse transcribing viruses, but you could also click on Baltimore Scheme and sort them by that. If you click on any of these divisions, like double-stranded RNAviruses, you get a list of all the known viruses, and you can click again and get pictures of the viruses. You can get pictures of the genomes. You can get examples of some of the viruses and the hosts that they have and the diseases that they cause. And if you're really interested, you can click on reference strains, and it'll go right to the sequence of the whole genome. Now, this is, of course, if you're interested in finding out more about a particular virus, it's a great site where you can do that. It also puts the Baltimore Scheme, I think, in nice perspective. Now, what do these genomes encode? As we go through this course, one of our goals is going to be to figure out how the genome is expressed and replicated. And so some of the information is important for replication of the viral genome. Every viral genome has to encode at least one protein that's involved in replication. But as you'll see, as genome sizes get bigger, some of them encode many more. They have to encode proteins to assemble and package the genome into a particle. So packaging means putting the genome in a particle, and they have to make the proteins that make up the particle as well. There are proteins involved in regulating the infectious cycle. Not everything happens at once. There is a temporal control of gene expression, and there are proteins for that. Then there are proteins that modulate host defenses. These are really interesting. This is not something we knew about more than 30 or 40 years ago. Every virus on the planet has to encode at least one protein that antagonized host defenses. Otherwise, it wouldn't be here. We have such good defense systems that if viruses didn't encode antagonists, they would be wiped out. But they do, and there's this arms race that we talk about. We will talk about that much later. And then they must encode gene products that involve spread to other cells and hosts. And now remember, this doesn't only mean protein. For many years, we thought proteins were the end-all and be-all of life, but now we know there are regulatory RNAs of all sorts, small RNAs that are made, and in fact, most of the genome is not protein coding, and those are encoded by viruses as well, and we'll talk about some of those. Some things that are not contained in genomes, which is really interesting to think about. As I've mentioned, no genes encoding a complete protein synthesis machinery. Now, some of the big viruses that were recently discovered, some of these giant viruses do encode some amino acyl tRNA synthetases. They encode some initiation factors. They encode even tRNAs, but not a complete set. We don't know why these viruses have a couple of tRNA genes, a couple of synthetase genes. People are still working on understanding. Maybe they infect a particular kind of cell, and producing a viral tRNA would help them replicate better. Nobody's done that experiment, but it's a possibility. There are no genes encoding proteins involved in energy production or membrane biosynthesis. So no virus can make ATP. No virus can make lipids to make a membrane. So viruses are dependent on cells for energy, and they're dependent on cells for membranes. A number of viruses do incorporate membranes into their particles, and they're dependent on the cell for that. There are no centromeres or telomeres that you find in your chromosomes, all right? Centromeres is ways of organizing them. Telomeres, the ends of our chromosomes that perform a clock-type mechanism. As you age, you're losing sequences at the ends of your chromosomes. Viruses don't have those. But, anything I say in any given year could change next year. When I first started teaching this course, they didn't have this first line in here. I used to say, no virus makes any part of the translation machinery. Now, we discovered giant viruses that do encode some of these genes. And it could be that some of these other genes will be found at some point. In fact, when we sequence the environment and look for viral genes, 90% of them are novel. We don't know what they are. We have no clue what these proteins look like because we haven't seen them before. So it could be in the future we'll find some of these genes. So I thought you might be interested in looking at the extremes of virology, the biggest and the smallest. These are the largest known viral genomes as of today. The biggest one so far is a virus called Pandoravirus selinus, and it's 2.4 million bases of double-stranded DNA. This is huge, absolutely huge. When this was discovered, the biggest viral DNAs were way less than 600,000, probably about 250,000 bases in length. So this is huge. And this encodes for 2,500 proteins. enormous amount of proteins, and most of those we don't know, we don't recognize them, we don't know what they do. We can recognize polymerases and enzymes and helicases and so forth, but we can't recognize most of the proteins. We don't know what they do. Then we have a number of other viruses with smaller genomes with equally interesting names. Pandora was called this because when they first discovered it and sequenced the genome, they didn't know what most of the genes did, so they said it's like a Pandora's box. We're going to open it up and see what happens. Viruses were the first giant viruses discovered. They're called because that stands for microbe mimic. They were actually thought to be a bacteria when they were first discovered. In fact, they were discovered in a patient in France, and they were put in a freezer for 10 years because they were thought to be a bacterium and nobody bothered to study them. And then when they were pulled out later, it turned out to be viruses. And then finally, this one at the bottom, which is still pretty big, encodes a lot of proteins, Pithovirus sibericum. This was recovered from a 35,000-year-old ice core taken from Siberia. So, people go into Siberia and they drill down and they pull out these long cores, and you can tell what's been going on in these areas by looking at what's in each core. It turns out that this virus was taken out of a 35,000-year-old piece of this ice core. They thawed it in the lab and outgrew this virus. So they can last for many years if they're frozen. Smallest known viral genomes. Now, at the top is what I call a viroid. It's not a virus because it's just a piece of nucleic acid. It's a piece of RNA, 120 bases long. It doesn't encode any proteins. And this is one of the relics from the RNA world. It's not a virus because a virus typically encodes something, but these are all artificial definitions anyway that we make. So I included in this course, and we're going to talk about viroids later on. So they don't encode proteins. There are also some larger virus-like particles called satellites, also don't encode any protein. Satellites are dependent on a helper virus to co-infect the cell and let them replicate. Viroids don't need any helper virus. And again, we'll talk about these near the end of the course. Then we have a human satellite called hepatitis delta, 1,700 bases, encoding one protein. And then here we have the first autonomous virus, which means it can get into a cell and replicate on its own. Circovirus, 1,700 bases of circular single-stranded DNA encoding two proteins. And then you can see they get slowly bigger and bigger and encoding more and more proteins. But I mean, 1, 2, 4 proteins, it's amazing compared to the 2,500 of the biggest viruses that we know of. And so these represent the minimum numbers of proteins that a virus would need to propagate. And you could predict very easily that these small viruses depend a lot on the host cell. All viruses, of course, depend on the host cell, but some more than others, as we'll see. What information may be encoded in a viral genome? Gene products that catalyze membrane biosynthesis, gene products that catalyze energy production, complete protein synthesis systems, centromeres or telomeres, or enzymes to replicate the viral genome. The answer is E. What information may be encoded? Enzymes to replicate the viral genome. None of these other ones, of course, are correct. They don't have genes that catalyze membrane biosynthesis or make energy. They don't have complete energy protein synthesis systems, and they don't have centromeres or telomeres. Now, let's talk about viral DNA and RNA genomes a bit separately. Viral DNA genomes, of course, look like our genomes. They emulate the host. So a lot of things that they do are similar to how we deal with DNA in our cells. And in fact, study of DNA viruses led the way in understanding how our genomes replicate. But there are differences. For example, almost all viral DNA genomes are not like our chromosomes. Our chromosomes, as you know, are chromatinized. The DNA is wrapped up around nucleosomes in a very higher order structure, and whether the nucleosome is tight or loose depends on whether it can be transcribed or not. So viral DNA genomes do not look like nucleosomes in the virus particle at all, except for one family of viruses. However, when viral DNAs get in the nucleus, which most of them do, they do become chromatinized. They become wrapped in nucleosomes, but in the particle they are not chromatinized. And as we will see as we talk about DNA virus replication, viruses have evolved tricks that are different from what the host does in order to get their DNA replicated preferentially. When you think about it, a virus enters a cell, the cell is replicating. Why should the viral DNA be replicated? It's one molecule coming into a huge chromosomal nucleus. Well, there are tricks that have evolved to allow the virus to do that. Now, here are some viruses with double-stranded DNA genomes, and these are, again, examples that we're going to talk about in this course in terms of replication and pathogenesis. The adenoviruses, which you will remember because they have this very unusual Sputnik-like appearance. The herpes viruses, which are membrane-encased virus particles. All of you have most of the known herpes viruses in you right now. Papillomaviruses. These are viruses that cause warts on your hands, on your feet, but they can also cause anogenital warts, which lead to cancer. And we'll talk a lot about them. Polyomaviruses are related to the papillomas. They're also small double-stranded DNA containing viruses, which have played a huge history in our understanding of how viruses cause cancer. And then this big one down here is a pox virus. Smallpox virus looks like this. These are huge, what we thought were huge viruses, 250,000 base pair genomes, but it turned out they weren't very big at all. So those are the double-stranded DNA genomes. How do they replicate? As you know, double-stranded DNA can go directly to mRNA. So when these viruses enter the cell, the first step can be transcription of that double-stranded DNA genome to make mRNA. The mRNA, of course, encodes for proteins. Now, in general, you will see that there is always at least one viral protein that has to be made in an infected cell before DNA replication can occur. That's an important rule to remember. So early in infection, some proteins are made to facilitate DNA replication. We'll talk about how the DNA of these viral genomes is replicated. And then in the end, the DNA and the proteins come together to make new virus particles. So we have two generally different types of viruses with double-stranded DNA genomes. Those in which the genomes are copied by the host DNA polymerase. The DNA polymerase that copies your DNA copies the DNA of these viruses. These tend to be rather small genomes. Polyomaviruses, papillomaviruses, 5 kilobases and 8 kilobases of circular double-stranded DNA. They don't have room to encode a DNA polymerase, so they use that of the host cell. The other viruses are genomes. Their genomes encode a DNA polymerase. They don't need the host DNA polymerase to replicate, although they do need, DNA replication doesn't involve just the DNA polymerase. There are lots of accessory proteins that are involved as well, and these viruses don't encode all of them, so they are dependent on the host for some of them. These include the adenoviruses, 36 to 48 kilobase pairs of double-stranded DNA. The herpes viruses are 120 to 220 kilobase pairs, and the pox viruses are even bigger. The point here is not, you don't need to remember the size. You just need to remember with the small genome, there's no room to encode a DNA polymerase. With a bigger genome, you can encode it. And that's what distinguishes these two classes of virus. Now we move to the gapped double-stranded DNA genomes. Hepatitis B virus is an example of that. It's a human virus that causes liver infection and liver cancer. Hundreds of millions of people on the planet are infected with this virus and will develop lethal liver cancer. The family name is Hepatnaviridae. The genome of this virus is a gapped double-stranded DNA molecule. So it's partially double-stranded DNA. Part of it is single-stranded, the negative strand there. It also has a protein attached to the 5' end, and it's got a piece of RNA attached to it as well. This genome cannot be transcribed. It cannot be transcribed to make mRNA. It enters the cell, and it's actually repaired by cellular repair enzymes. They remove the protein and the RNA, and they complete the double-stranded DNA genome. All right, so the host enzymes are doing this. And then you get a double-stranded DNA genome that is now the substrate for transcription of mRNAs. And of course, from there, viral proteins can be made that can be used to make new particles. Now, the way the genome replicates is very unusual here. Some of these mRNAs that are not used for translation are copied instead into first a minus DNA and then a double-stranded DNA molecule. That is done by the viral enzyme called reverse transcriptase. So two families of viruses that we're going to be talking about encode reverse transcriptase. And that enzyme name, of course, means that it takes RNA and makes DNA from it. It had always been thought that the flow of genetic information in life was DNA to RNA to protein. When this enzyme was discovered, it suggested that information could go from RNA to DNA, and that's why it was called reverse transcriptase. So hepatinoviruses take some of their messenger RNAs and make a double-stranded DNA from them, and that is what's packaged into the virus particle, actually double-stranded DNA. And why it's gapped and so forth, we'll learn about when we talk about the specific replication. Then we have viruses with single-stranded DNA genomes. These can be circular or linear. The circoviruses are rather small. These are viruses that are in all of us. TT virus, for example, is in everybody. They've done serological surveys. If I surveyed all of you, took some of your blood, I'd find antibodies to this virus probably in every one of you. But as far as we can tell, it doesn't do any harm. And it doesn't encode many proteins either. The parvoviruses are linear, single-stranded DNA molecules. These cause human diseases as well as diseases of pets, dogs, and cats. These viruses can package either strand of DNA, the minus or the plus strand. Doesn't matter, either can initiate the infection. But of course, a single strand cannot be transcribed. So when these minus or plus DNAs enter the cell actually repairs them and makes them double-stranded. The double-stranded DNA then can be transcribed, giving rise to viral proteins. The genome will replicate to give rise to single strands, and either can be packaged. And some of the details of this replication, again, we're going to talk about later. This is meant to be an overview for you. All right, our question is, which DNA genome on entry into the cell can be immediately copied into mRNA? Double-stranded DNA, gapped double-stranded DNA, circular single-stranded DNA, linear single-stranded DNA, or all of the above? Right, the answer is A, of course. Only double-stranded DNA can be immediately copied to mRNA. You got to remember that. It's a key, okay? Single-stranded cannot be immediately copied to mRNA. It has to be made double-stranded. Gap DNA cannot be immediately copied. Only double-stranded DNA. It's one of those things you just have to remember because it will make many things easier for you. All right, RNA genomes. A key point here is that cells do not encode an enzyme called RNA-dependent RNA polymerase, or RDRP. I'll abbreviate that often. RNA-dependent RNA polymerase. So we have DNA-dependent DNA polymerases that replicate our genomes, replicate genomes of DNA viruses. For RNA viruses, the enzyme is called an RNA-dependent RNA polymerase, and cells do not encode them. What does that mean? Every RNA virus on the planet has to encode an RNA polymerase. It's not like the DNA viruses where some can encode a DNA polymerase and some can use that of the host cell. The host cell does not have an RNA polymerase. So every RNA virus genome has to encode one. Of course, the viroids don't encode anything. You may be asking, well, what replicates them? Well, we will find out later. So RNA virus genomes encode an RNA polymerase, and that RNA polymerase copies the genome, and it makes mRNA from the genome. So it has two functions. For DNA viruses, those functions, replication and transcription, are carried out by two different enzymes. Replication of DNA is carried out by DNA polymerase. Transcription of DNA to form mRNA is carried out by DNA-dependent RNA polymerase. For viruses with RNA genomes, it's the same catalytic enzyme that carries out genome replication and mRNA synthesis. This is a very important point because it will help you understand how RNA viruses work. Because again, this together with the Baltimore Scheme, you can figure out the pathway of genetic information. So let's take a look at the different categories of RNA virus genome. We have double-stranded RNA genomes. This happens to be one, the reoviruses, and a virus within them is called rotavirus, causes human gastroenteritis. If you've ever suddenly gotten ill with vomiting and diarrhea, by the way, vomiting and diarrhea, we call it two-bucket disease, because you need a bucket at either end to collect the fluid. And not everybody can do that. You know, bacteria can do a one-bucket disease, but viruses can do two-bucket disease. Rotaviruses, the other one is the neurovirus. The cruise ship virus will also give you a two-bucket disease as well. These viruses have double-stranded RNA genomes in pieces, 10 or 11 different segments. Some of them have fewer, but they're double-stranded. And the key here is, could you make protein from a double-stranded RNA? Given that there's a plus mRNA in there. Can you translate this? No, you can't. Only mRNA, single-stranded mRNA, can be translated. So The virus particles have double-stranded RNA in them, and they get into a cell, and they sit in the cytoplasm. There's no enzyme that could make mRNA from them. So the virus has to carry in the particle an enzyme to make mRNA from the double-stranded genome. That's something you could sort out on your own if you just thought about it a bit, because the cell can't copy mRNA from these double-stranded RNA, so the virus brings in an enzyme. It makes mRNA from this double-stranded RNA. The mRNA can encode protein. It can lead to replication of the genome and the assembly of new virus particles. We'll talk about this in more detail, but the point here is that a double-stranded RNA genome contains an mRNA, but it can't be accessed. It has to have fresh mRNA produced from it. And we have the viruses that make a lot of sense to me, the plus-stranded RNA viruses. There are tons of them. You know, in fact, overall, most of the viruses that cause disease are RNA viruses. I'm not sure why that is, but we'll talk about it later. Here are the plus-stranded, single-stranded RNA viruses. Picornavirus is the family. It includes polio and rhinoviruses. They're small RNA viruses. Caliciviruses includes Norwalk virus, the cruise ship virus, which causes gastroenteritis. The coronavirus, the agents of SARS and MERS in the Middle East. And then the flavies, yellow fever, West Nile, hepatitis C, and more recently Zika virus. We'll talk about those. And finally, Togaviridae. These are viruses including rubella virus and equine encephalitis virus. Now, these all carry in their particles a plus-stranded RNA. And because it is plus-stranded and can be translated as soon as it gets in the cell, the virus does not have to have an enzyme in it. There's no enzyme in any of these virus particles because it's not needed. What happens is the RNA gets into the cell, can be immediately translated. The proteins can then lead to genome replication and the assembly of virus particles. So when I mean genome replication, I mean producing more plus-stranded RNAs. To do that, you have to go through a negative strand copy. You make a copy of the plus, that's a minus strand. The minus strand exists only as a template to make more plus strands. These are what the genomes of these viruses look like. Here, the coronaviruses, they are the biggest RNA genomes we know of, 33 kilobases long. They don't get any bigger than that. There are constraints. Flaviviruses, including Zika virus, 10 to 12 kilobases. Picornas, slightly shorter, and togas, 10 to 13. You can see these viral genomes all have the green part. By the way, I color code all of the genomes in these slides. This kind of Kelly green is plus strand. This brown olive color is minus strand RNA. And the same thing for DNA, plus and minus are two different colors. So these are all plus-stranded. And then the yellow is 5' and 3' non-coding regions, doesn't code for protein. And you can see the structure of these RNAs are different. As you probably know, an mRNA in a cell needs to have a poly A tail in order to be efficiently translated. Some of these viruses have that, but others do not, like the flaviviruses. We'll talk about how they're translated in the absence of a poly A tail. You may also know that a cap at the 5' end is needed for efficient translation. Many of these genomes have a cap, but not all of them. You can see there's a protein here at the 5' end of these genomes. We'll talk about how that works as well. Now, we have this one unusual family, the retroviruses, where they have plus-stranded RNA in their particle, but when that gets into the cell, it is not translated because it's shielded in proteins, so the ribosomes can't get to the RNA. There are two human pathogens in this family, HIV, human immunodeficiency virus, and human T lymphotropic virus. And we'll have a lecture for HIV on its own because it's a very special virus, the only virus that gets its own lecture in this course. These viruses have, as I say, plus-stranded RNA genomes, 7 to 10 kilobases in length. They have a cap at the 5' end, poly A tail. But as they're packaged in the virus particle, they're not translated when that RNA comes into the cell. Rather, there's a reverse transcriptase in the particle that copies the RNA into DNA right in the cytoplasm. First it makes minus DNA, then it makes double-stranded DNA. And now that, as you may guess, could be a substrate for mRNA synthesis. But in order to be transcribed, the DNA gets in the nucleus and it integrates into the chromosomal DNA of the cell. That's how retroviruses replicate. The integrated copy of the retroviral DNA is called a provirus. So that's a definition. It's integrated into the genome. It's a provirus, and when it's integrated there, it is transcribed by cellular polymerase to make messenger RNA. A retrovirus encodes a reverse transcriptase, a different enzyme. So every RNA virus has to encode some kind of enzyme to replicate its genome or make mRNA. And the slight difference are the retroviruses, which don't encode an RNA-dependent RNA polymerase, but an RNA-dependent DNA polymerase, right? But they do not encode an enzyme that can make mRNA. So the RNA-dependent RNA polymerases of other RNA viruses can make mRNA, but retroviruses cannot. So they are dependent on the host cell to make their mRNAs. And the mRNAs give rise to protein. The mRNAs are also packaged in new virus particles. So the genome of retroviruses is an RNA. And now remember, I told you the genome of hepatinoviruses is DNA, yet both viruses undergo reverse transcription. And why that's so, we'll talk about later on. These are really unusual viruses because of this provirus. It can integrate into your DNA, and that means it can stay around as long as the cell lives. And if this happens to integrate into your germline, you will pass these viruses on to your kids. Now, fortunately, HIV does not integrate into the germline, cannot infect germ cells. So there's no vertical transmission of HIV. If there were, it would be even worse situation than we are in now. Right, minus-stranded RNA viruses, there are a whole bunch of them as well. Paramyxoviruses, measles virus, and mumps virus, which you don't get anymore because you've been immunized. When I was a kid, every kid would get measles and mumps. You would know because they wouldn't be in school. They couldn't come to school. And they would get a rash disease or swollen salivary glands from mumps virus. No more. Rabies virus here, bullet-shaped virus, almost 100% lethal. It's the only virus that is 100% lethal. If you don't get vaccinated, you will die of rabies virus infection. Ebola viruses. Everyone's afraid of Ebola virus, but they're not 100% lethal like rabies virus is. There's no vaccine yet, but and they're quite serious, but I think rabies is the more serious one. Lots of rabies still in the world. Influenza viruses are called orthomyxoviridae and arenaviridae, Lassa virus. So these are all negative sense RNA viruses. Now, Lassa virus was discovered in the 60s, and there's a great book that recounts that whole story of its discovery. It's called Fever. And I bring this up for two reasons. First, part of the story took place at Columbia Medical Center. It's really interesting. And I read this. I graduated from college, and I didn't know what I wanted to do with my life. So I read this book. I was working as a lab technician. I read this book, and I decided I wanted to be a virologist. So I'm here because of this book. So I feel an obligation to show it to you. There are two different kinds of viruses with negative strand genomes, ones that have the genome of a single molecule of RNA and some that are segmented. And the picture I just showed you was a mix of both. You can have segmented genomes like influenza viruses, or you can have non-segmented genomes like the paramyxoviruses and the rabies virus. But the overall strategy is the same. It doesn't matter whether it's in pieces or not in pieces. And by the way, when I say segmented, most of the time, all the segments are in one particle. But there are examples where some segments are in one particle and the rest are in another. And that would be a virus where you need two to infect a cell to get infection. So the viruses with negative strand RNA, this RNA comes in the cell. It cannot be translated. The cell cannot copy it. It doesn't have an RNA-dependent RNA polymerase. So again, you can go through this logic. The virus particle must have an enzyme in it so that when this RNA gets in the cell, it can be copied to mRNA. And in fact, all these negative strand viruses carry an RNA-dependent RNA polymerase in the virus particle. Of course, they also encode for the polymerase in their genomes so that they can make more of it in the infected cell. So when these viral RNAs get in the cell, they're copied immediately to plus RNA by the viral enzyme in the particle. That can be translated into protein. The protein then can mediate the replication of the negative strand genome to make more minus RNAs and eventually the formation of particles. And as I said, these genomes can be in pieces, like for influenza virus, or they can be non-segmented. Now, the presence of a segmented genome has important consequences for the genetics of the virus. When you have a segmented genome, you can undergo something called reassortment. This is a specific term. Now, you may be familiar with recombination, where two DNA molecules will exchange sequences. But this is different. This is where two viruses with segmented genomes can exchange segments, entire segments. And that's illustrated here. We have two different influenza viruses, L and M. L has red segments, and M has blue segments. So the influenza genome is composed of 8 segments. When these both infect the cell, now in nature, there are probably lots of co-infections going on, and the likelihood that you can get a co-infection of an animal, say, with 2 different viruses is very high. And if those 2 viruses infect the cell, as the RNAs are replicating, you have both blue and red RNAs being made, and then the viruses that are eventually produced can be the parental L or M virus, the red or the blue, or they can be reassortants. So here's an R3 reassortant where the second segment is red. All the others are blue. They come from the M parent, and the red segment comes from the L. So this is reassortment. And the reason why this is important is, first of all, reassortment is a high-frequency event. It's much higher than recombination, which depends on either breaking of sequences or polymerases changing from one strand to another. And for viruses with segmented genomes, that gives rise to more variation. So for example, influenza virus, periodically brand new strains emerge to which none of us have immunity. And they emerge because human and animal strains undergo reassortment in animals out there in nature. And those strains, so basically reassortment can create new diversity to make more strains to infect people. So we're going to come back to that later and explore the consequences. Now there's a family of viruses with ambisense RNA genomes. These are very unusual. The strands of RNA have both plus and minus polarity. So you may say, how can you tell? Because if you sequenced it, it would be hard to tell just by looking at it. It's just RNA. But what you can do is look at the genome of this virus. These are arenaviruses like Lassa virus. The genome is a mix of plus and minus, because here at the 5' end, you have coding sequence. You can look at the sequence and translate it, and it makes one of the viral proteins, the glycoprotein. Then there's a stop codon, and the rest of the genome is not translatable because it's in the negative sense. It encodes a second protein. And so these viruses have typically multiple genomes in their particles, can be up to 3, and each of them has a plus and a minus RNA fusion. Now you may wonder, how do you access this information? Well, this RNA is in the particle, and you may think that this first part of the RNA could be translated because it's capped and it's a plus strand, but it's not. What happens is these viruses have an RNA polymerase in the virus particle. The RNA polymerase makes a small mRNA from the 3' end of these genomic RNAs, so that's shown right here, npRNA, and then that is translated into protein. So the way this coding sequence is accessed is to make an mRNA, which is translated. The genome is then copied, and we call it a genome and an antigenome, because it wouldn't be right to call it a plus and a minus because it's a hybrid, again. Make an antigenome, and now you can make an mRNA from this portion of the antigenome that will encode the glycoprotein mRNA, and that will be translated into protein. Very unusual strategy. Again, these are ambisense. They can have both characters of plus and minus on the RNAs. Now, I've been showing you RNAs as little squiggles of different colors, but that probably doesn't look anything like an RNA does in a virus particle. It has extensive secondary and tertiary structures. So here on the upper left is how I've been drawing most of these viral genomes, but people have been studying these in a variety of ways for many years, and it's quite clear that they look different. They have a lot of structures. And here at the bottom is an RNA of a plant virus where the interactions, the long-range interactions, have been dissected. So you can see, first of all, there are these stem loop structures throughout the RNA. These are areas where the RNA base pairs with itself and makes a structure, a very highly stable structure depending on the bases involved. And RNA viruses typically are loaded with these. You can see this part of the RNA has multiple stem loops that go on and on and on throughout the genome. And these stem loops are important. They interact with proteins and they mediate many of the basic functions of the viral genome. But there's also long-range interactions. So for example, this red portion at the left here is known to base pair with this red portion far downstream. And the two purple portions are known to interact as well. The orange portions interact. So think of RNA not as a line, but as a ball all wrapped up with all this kind of secondary and tertiary structure. And then it becomes even more interesting to wonder how an enzyme would even get through this. How does a ribosome get through to translate it? How does a polymerase get through to translate? These are really interesting questions that people are trying to work on. All right, our last question for today is which statement about viral RNA genomes is correct? Plus single-stranded RNA genomes may be translated to make protein. Double-stranded RNA genomes can be directly translated to make protein. Plus single-stranded RNA virus replication cycles do not require a minus strand intermediate. RNA genomes can be copied by host cell RNA-dependent RNA polymerases, and all of the above. Well, the answer is A, plus single-stranded RNA genomes may be translated. They don't have to be, right? The retroviral plus mRNAs are not translated. But all the others aren't correct. Double-stranded genomes can't be translated. It's an important point. The mRNA is buried in there. It has to be transcribed out. Plus single-stranded RNA virus replication cycles do require a minus strand intermediate. That's how you make more plus strands, by making a minus strand. RNA genomes can be copied by host RNA polymerases. There are no such enzymes. There are no host RNA-dependent RNA polymerases. And of course, it can't be all of the above. All right, now I just want to end by talking a little bit about how we manipulate viruses, to study them and to use them. Now, when the plaque assay was developed, that was how we did viral genetics. We would take a stock of virus, incubate it with a mutagen, UV light or some mutagenic chemical, and then we would plaque out the resulting viruses and pick individual plaques and study individual mutants. So the early history of virology is full of this, making mutants and studying them by plaque assay. But that all changed 30, 35 years ago with the development of recombinant DNA methodologies and the ability to make a DNA copy of every virus genome, and for that genome to be infectious. And this is what we do now. This is how we put mutations in the viral genome. It's the infectious DNA clone. We introduce DNA into cells by transfection. And this is a modern validation of the Hershey-Chase experiment. You take just the viral genome, whether it be RNA or DNA, you make a DNA copy, you put it in a bacterial plasmid so you can make lots of it, and then you take that recombinant DNA, you put it in cells, and out comes virus. The DNA is the genetic material. And why is this so good? Because you can manipulate it. You can make mutations of all sorts. You can delete, insert. You can make recombinants. You could add genes into viruses or take them away, depending on what you want to do. Recombinant DNA makes that possible. Of course, with RNA viruses, you can't do this at all because the RNA genome is not manipulable. So if you have a DNA copy that is infectious, you can now make all kinds of changes. The last lecture in this course will be about viral gene therapy. We're going to talk about modifying viruses to be vaccines, to cure cancer, to deliver therapeutic proteins to people who lack them. It is the future. In 10, 20 years, we're going to be curing cancer, we're going to be curing genetic diseases, we're going to be doing all sorts of things by using virus vectors to deliver things to people. The whole field is made possible by having infectious DNA clones of different viruses. So let me just tell you a couple of examples of how this works. First of all, the word transfection is the production of virus after transformation of cells by viral DNA. It was first done with a bacteriophage, and it means transformation, infection. So you transfect DNA into cells and you get virus out. Now, nowadays, people use transfection to mean just putting DNA of any kind into cells. It's not really quite correct. It was meant originally to indicate viral DNA. But the cause is lost. We have to use transfection for everything. So here's an example of poliovirus, where we know if we infect cells with polio, we get viral replication, we get the production of infectious particles. Polio is a plus-strand viral RNA. In fact, if you extract the RNA from a particle and put it into cells by transfection, it will initiate a replication cycle. Think about it. The plus-strand RNA is a message. It gets into cells. The viral proteins are made. You start an infectious cycle. It's basically like infecting a cell, except you're not using the virus particle. If you instead make a DNA copy of polio RNA and clone it in a plasmid in bacteria and grow it up, you can take that cloned DNA, transfect it into cells, and you'll get virus out as well. So that's what I mean by infectious cloned viral DNA. You can manipulate this DNA and get out any virus you want. For example, down at Duke, people are using a modified polio virus to cure gliomas. They inject the virus directly into the tumor of a patient with a glioma, which you may know is pretty much a fatal tumor, and they've had some success. Not complete, but it's prolonging people's lives, and presumably if they work on it, they can get it to be even better. That's what you can do with having a recombinant DNA copy of a viral genome. We can do this with influenza virus as well, which has 8 different RNA segments. What we do is take each RNA segment, we make a separate plasmid encoding each RNA segment. We put 8 plasmids into cells, and we can recover infectious influenza virus. Now, influenza is one of those viruses that we only have the virus since 1933 or so. But you may remember in 1918, there was a huge global pandemic of influenza virus that killed many, many millions of people. Well, we didn't have virus isolated. It wasn't discovered in 1918. But what was done in 2005, so the Army, of course, was involved in World War I at the time. They had lots of people in the Army dying of influenza, and many of them, they took samples of their lungs, put them in paraffin, formalin, preserved them in paraffin, and stored them away. Well, in 2005, investigators extracted RNA from those formalin-fixed lung tissue samples, and they got some influenza virus sequence. They also went up to Alaska and found a victim of 1918 flu who had been buried since then and kept frozen. They opened the grave, they did a biopsy of the lung, and they recovered influenza RNA. They put together all these sequences. They put together the complete sequences of all 8 segments. They made DNA copies of them, and they recovered this virus. So the 1918 influenza virus that killed many, many millions of people, we didn't have it, but modern technology allowed us to make it. All we needed to do was get the sequence. Now, a lot of people did not like this experiment to be done because they said it was dangerous. We're going to release this virus which killed many, many, many people. However, having the virus allows us to make a vaccine, to know that antivirals will inhibit in case this virus ever returned. And we're beginning to study the biology of this virus and why it was so severe. All the work on this kind of virus, of course, is done under very high containment called BSL-4, the kind of laboratory containment where you have to wear pressurized suits and so forth. But nevertheless, there are many people out there, a lot of them politicians, who don't like this kind of work. So now we have regulations to govern what we can do and what we can't do. And I want to just end up talking a little bit about this. There's an advisory committee of the US government, National Science Advisory Board for Biosecurity, NSABB. And this is supposed to provide oversight and guidance regarding what we call dual-use research. So if you do research on influenza, 1918 influenza virus, you may have a good goal in mind, making a vaccine or something. But it could also be used for nefarious purposes. Someone could make a bioweapon out of it. So this committee is supposed to look at any proposed research involving dangerous viruses and say, maybe you shouldn't do this, or maybe you should do it under this sort of containment. So they advise on all federally supported research. So if you do research on money provided by a company or provided from overseas, you're not subject to these regulations. Now, very recently, there was a moratorium on certain kinds of experiments using these viruses imposed in part by work of this committee. It was just raised. But going forward, this whole area is called synthetic virology. You've heard of synthetic biology, where we want to make organisms from scratch. Well, we've been able to do this with viruses for a long time. And people are basically scared of it. I think they're scared because they don't understand it. I think there's always a good part of this kind of, it's always meant for good, this kind of research on dangerous pathogens. You just have to make sure you do it under the right conditions. And there are lots of regulations in place to make sure that happens. But what I fear is that a lot of our good discoveries come from dangerous experiments. And if we simply have a knee-jerk reaction, no, we shouldn't be doing this because it's too dangerous, that's going to limit scientific progress. So I'm a very big proponent of regulated experiments, but allowing them to be going forward under the right conditions.